All right, good morning, good afternoon, good evening. This is Dr. Kaz here in a secret location, my bunker, somewhere in an undisclosed place in South Carolina. I'm going to be wrapping up chapter 4 with you folks today. And we were talking about membrane transport with the plasma membrane. We just finished up talking about the passive processes of both diffusion and osmosis. And now we're going to transition into the active processes. Um, Big difference between active and passive processes. Active processes require energy, ATP, and passive processes do not. Passive processes will also move things down their concentration gradient. Okay? Active processes are different. They will move things against or up their concentration gradient. Okay, so when we talk about the active processes, we divide them into the active transport and vesicular transport. Think of vesicular transport as moving something in a package, kind of like what UPS does or Amazon. Okay, so that's going to be our vesicular transport. All right, this will require energy and it will move things. All right, and in this case, that will be our solute against its concentration gradient. So much like we'll be moving our boulder and pushing it up a hill, and so therefore we're pushing that boulder up the hill, it's going to cost us some energy. Okay. Okay. Let us talk about, all right, a couple different types of active processes here. All right, I'd like to talk about ion pumps. We're going to be addressing different types of ion pumps this semester. Mainly we'll talk about the sodium-potassium ion pump when we get into the neurons, but think of it literally like a pump, okay? And these transport proteins are going to be pumping the solute, all right, into the cell or out of the cell. And this could be happening in one direction or it could be happening simultaneously. What I mean by simultaneously, the pump could move two different solutes in two different directions. All right, so it can pump one, like for the example for the sodium-potassium pump, it'll pump sodium out and then of the cell, and then when it's done pumping sodium out, it will pump potassium in. Okay, but its big job is to help to maintain that concentration gradient or that concentration of those ions or solutes, mainly ions. All right, in the cell and outside the cell. So we see an ion pump in red blood cells in the erythrocytes for calcium. And calcium gets pumped, all right, out of the cell, okay? Because when calcium penetrates the cell, and if it accumulates, it can cause the cell to become rigid, and you don't want that. Imagine a red blood cell trying to squeeze in between two simple squamous epithelial cells in your capillary, or what we call the endothelium. And if that cell is rigid, it is likely to become damaged. And we can't have that, all right? Because then we've destroyed something that, a structure that is crucial to cellular respiration because it carries oxygen and carbon dioxide. This picture here depicts, all right, the sodium potassium ion pump. All right, you can see here's the pump in the plasma membrane. Okay, ATP will attach itself onto the pump. The pump will then open up and release sodium that was inside the cell and push it outside the cell. All right, and keep in mind there's a higher concentration of sodium outside the cell. All right, because as you know, with nature, and the diffusion and whatnot, and we're always trying to maintain and reach equilibrium, okay, sodium wants to come into the cell, but this pump is pushing it out of the cell. Okay, so as that occurs, as the sodium is getting pushed out of the cell, right, our ATP molecule, right, breaks down into ADP and one inorganic phosphate molecule. All right, and as that inorganic phosphate molecule detaches, all right, potassium has moved into the cell, into the pump, all right, and then that pump is going to return to its original configuration, just like this, and the potassium enters into the cell. Again, against its concentration gradient, because there's more potassium inside the cell. Banana floating in the ocean. More on that in chapter 12. Okay, so that's the sodium potassium ion pump. All right, so keep this in mind. All right, when we're dealing with the sodium potassium ion pump, it is an exchange type of pump, meaning we're going to exchange 3 sodium and for 2 potassium. And we will move these substances, these ions, against their concentration gradient. All right, it will cost us 1 ATP, but that 1 ATP gets us 2 potassium coming into the cell and 3 sodium moving out of the cell. All right, so that is one type of transport membrane. All right, now we're going to talk about secondary active transport. Same type of situation. We are going to be moving substances against or up their concentration gradient. The nice thing about this phenomena, right, when you're moving things, you are generating, we talked about this in a previous class, you are generating what we call kinetic energy. Okay, this energy is generated from movement. Okay, so in secondary active transport, right, we're going to be dealing again with 2 substances. Okay, whereas when we move one substance, substance A, all right, it will move down its concentration gradient. Okay, so like the diffusion phenomena of something moving down its concentration gradient, going from an area of high concentration to an area of low concentration. All right, well, substance A, as it's moving down its concentration gradient, it will generate kinetic energy. And therefore, we can move substance B, all right, up its concentration gradient, okay, with that energy. So there's 2 types. We have what we call symport and antiport. All right, symport is when we move 2 substances in the same direction. So same, okay? And then antiport is going to be in opposite directions. So symport we see in the thyroid, okay, gland, as we are trying to get iodine into the thyroid, the follicular cells there. All right, it kind of piggybacks on sodium. So, and it moves through, through this active transport mechanism, because sodium is going to enter into the cell because it's moving down its concentration gradient. But as it does so, okay, iodine is going to enter in with it. All right, but in that situation, the iodine is going to be moving up its concentration gradient. Believe it or not, those follicular cells have a ton of iodine already inside them, and we're just trying to get more in there. All right, and then of course with antiport, you have the 2 substances moving in opposite directions. As one enters in, and one will exit the cell. Okay, again against its concentration gradient. So we were talking about active transport processes. Now I want to get into the vesicular transport. Okay, and one of these here we're going to talk about is exocytosis. Now, way back in the beginning of this chapter, we were talking about proteins being made, okay? And those proteins can be made in the cytosol or cytoplasm, and they can be made in the rough endoplasmic reticulum. If they're made in the rough endoplasmic reticulum, they're going to get packaged up and sent to the Golgi apparatus. And that Golgi apparatus, that warehouse of the cell, is going to modify it and package it up nicely to be sent to the plasma membrane. All right. It has 3 options when it gets packaged up from the Golgi apparatus. All right, it can get sent to the plasma membrane, and the contents thereof, all right, that secretory vesicle can be incorporated into the plasma membrane if the plasma membrane was damaged, or if the cell is growing, or what have you. All right, or if we have to upregulate certain pumps, like we were talking about that glucose pump. Okay, not pump, excuse me, that glucose transporter there. Okay, and then a second option would be if, say, it's a beta cell and we're making insulin and we want to get the insulin to the bloodstream. All right, that second option with the vesicular transport, that secretory vesicle will then, same thing, it will fuse with, all right, our plasma membrane like it's depicted here in this picture here. All right, it'll fuse with the plasma membrane. It'll start to open up and release its contents, all right, into the interstitial fluid. And so it'll be released from the cell. And the third option when we're dealing with the Golgi apparatus, those proteins will be enzymes, the digestive enzymes for a lysosome, and those digestive enzymes will be incorporated into a lysosome. All right, so that's exocytosis. So think of exo, exiting. We're going to exit the cell with our stuff. All right, now we're going to deal with endocytosis. Okay, and endocytosis is now the cell is sitting there, and there's something outside the cell, and it wants it. Because remember, small particles that are nonpolar can diffuse directly through the plasma membrane. Small particles that are polar will need a little help. All right, we'll have those transport proteins to help out, like channels and whatnot. But what about the large stuff, the big stuff? No problem. We have the fix for that. All right, we have our endocytosis. So we have something that's large on the outside, maybe some big lipid droplets or whatnot, and they are on the external side of the cell. And so basically, all right, endocytosis will be the opposite of exocytosis. Okay, so the cell is going to form, all right, and I'll show you a picture here in a second, is going to form, all right, these pseudopod-like structures and kind of wrap itself around whatever that substance is and draw it inside. If you've ever seen an amoeba, a single cell organism, do its thing, it's very similar. In fact, that's how amoebas pretty much get a lot of their nutrients, is through that process there. Okay, so let's talk about, there's a couple different types, all right, of endocytosis. Phagocytosis, pinocytosis, and then receptor-mediated endocytosis. We're going to go through all three of those. So the first one, phagocytosis, is cell eating. Okay, and basically we have our large molecule on the outside of the cell. The cell creates these pseudopods, okay, and it extends itself around that particle and engulfs it. And then it kind of creates a sac in that process, and it pinches off that sac, and it pulls it to the internal environment of the cell, and then it'll fuse a lysosome with it. This is actually how we make, well, forget that, strike that. So that lysosome will then dump its digestive enzymes into that vesicle there, and it will break down whatever's in there. All right, and one of the more famous cells that do this is going to be the macrophage. Macrophage is a white blood cell. You get to learn more about that in 211. All right, but the macrophage, if it identifies something outside the cell, okay, say it's a bacteria, and it'll do this process, and then it'll actually digest and break down that bacteria. And then it's kind of weird because a macrophage is what we call an APC, and that means it's an antigen-presenting cell. And it it breaks down whatever that pathogen was, and then it'll take pieces of it and stick it on its own plasma membrane. Kind of interesting because it digests the thing and then sticks it all over its body. And then it wanders around looking for other white blood cells. And it says, will you look at this? Is this supposed to be here? And if it's a foreign protein, like in this case, a bacteria, then the white blood cell says, oh, heck no. And then it initiates the immune process. So it's kind of cool. All right. The second type of active process here, all right, for endocytosis is cell drinking. We have cell eating. Now we're going to have cell drinking. So in this case, instead of dealing with like a large solid molecule, now we're going to deal with maybe some lipid droplets or whatnot, all right, or some fluid, okay? So in this case, it won't be just one large vesicle. Now we'll make many small vesicles, all right? And the deal with this type of endocytosis is that it's non-specific, okay? Meaning the cell ain't picky. It just sees some stuff outside, all right? These droplets out there, and it says, I just want them. And I just need to get them in the inside, then we'll deal with it later. Okay, and that's what happens. Okay, so you have all these small little molecules, these droplets, usually lipid droplets, surrounds them, right? Smaller vesicles, pinches them off, draws them to the inside. Same process happens. Okay, and then finally, our last one is called receptor-mediated endocytosis. All right, in this case, all right, we are going to involve receptors. Okay, so we'll have some receptors on the outside of our cell, and we'll talk more about this when we talk about the metabolism of cholesterol. It's really a kind of a fascinating process here. All right, but as cholesterol is floating through our bloodstream, all right, it's actually stuck onto what we call LDL. And you may have heard of LDLs before. They have the nickname of bad cholesterol. Okay, and all of these LDLs are these proteins, these lipoproteins, all right, that sit on around the cholesterol here. All right, and they float around the body. Okay, and what happens is these cells will have specific receptors for these LDLs because the cell wants to internalize, all right, those lipoproteins. It might need the cholesterol for some sort of function. Maybe it's a cell that is going to metabolize or make hormones, because cholesterol is one of the ingredients for our hormones. And as we recall earlier on in this chapter, cholesterol is also important for our plasma membrane. Okay, it helps increase the integrity of the plasma membrane. So maybe we need to do that to repair a cell or just make a cell more hardy. So what will happen is we have these receptors on the outside of the cell. All right, these LDLs come floating by. All right, they make contact with the receptor, and then the cell, all right, will internalize or draw in that molecule. So I'll show you a nice picture of that. So here we have our molecules. Okay, excuse me, our molecules, and then we have our receptors, the little Y guy here. Okay, so our ligand, all right, or our LDL, makes contact with the receptor. All right, you can see we get enough of them, then the plasma membrane starts to fold in on itself. All right, with its LDLs stuck to the receptors. You might get a couple other things, except now on this picture here, you probably notice these green structures here. All right, that's a special molecule called clathrin. Okay, and when the vesicle pinches off, it has this clathrin coating, all right, that makes it easily identifiable to the cell as having these LDLs on the inside. All right, so we can facilitate whatever purpose that the cell has for these molecules. Okay, be it, like I said, for hormone synthesis or for cellular repair. Okay. All right, guess what? It's that time. We are now on to the very last part of the cell. Going way back to the beginning of this chapter, we talked about 3 parts to the cell. Okay, and we just got done talking about one of the parts, the plasma membrane. And we started off the chapter talking about the cytoplasm. And the third part of the cell is the nucleus. Okay, and this is the largest structure when you're looking at a cell, all right, using a microscope. This is the largest structure that you can see inside the cell. And I often refer to it as the control center. Okay, pretty much, all right, we assume that all the cells in the body have a nucleus. Well, that's not true in some situations. Most of the cells will have one nucleus. All right, some can have many, like we saw with the skeletal muscle cell. Even the cardiac cell can have 1 to 2 nuclei. All right, but all right, some cells don't have any. Okay, now in that situation, like we talked about briefly with I was talking about the keratinocytes in the skin, it loses its nuclei in the cell organelles through that process of keratinization, but that's essentially dead. All right, the erythrocytes, or red blood cell, they live for about 120 days. All right, and they do not have a nuclei. Okay, they do not. So as we saw when we were going over the tissues, all right, that the nuclei can mirror the shape of the cell. All right, we saw with the cuboidal cell, it was spherical, all right, meaning a cuboidal cell is relatively uniform. It's as tall as it is wide. Well, you know, that it has some uniformity to its shape. So does its nucleus, even though it's a sphere, obviously, but it's going to have uniformity to that. When we talk about the columnar cell, we saw that it's taller than it is wide, and its nucleus looks more oval, like an egg. But one of the most important facts, details about a nucleus is our DNA lives there. Our genetic makeup, the blueprints to us, are all inside our nuclei. Okay? And, you know, when we're making proteins or when we're undergoing cellular division, we're constantly accessing, you know, that information, that DNA information. Okay? And if you were to take a genetics course, you would go into it in a little bit more detail. We'll briefly talk about it in Bio 210 and 211, but you really can't appreciate the process of transcription and translation, all right, which is when we are either replicating DNA or when we are actually using that DNA to create a protein, and it's given us the instructions on how to do so. So if you ever do the next course and you go over that, it's fascinating stuff. Anyways, so when we're talking about our nucleus, it's similar to the plasma membrane, as the plasma membrane has provides the cell with its envelope that separates the internal cellular environment from the external cellular environment. The nucleus also has its own envelope. Okay, and it's similar in the fact that it's a phospholipid bilayer. It's a double phospholipid membrane. Okay, and so it keeps the internal contents of the nucleus separate from the cytoplasm. Okay, so we refer to that internal environment there as the nucleoplasm. Okay, but throughout the nuclear envelope there, all right, you have these little holes. All right, we call those nuclear pores. Okay. And so basically, all right, that allows, because if you recall, our ribosomes, and our ribosomes are these proteins that help to make proteins for protein synthesis and whatnot. All right, those ribosomes inside the nucleus. So we are going to send them, because remember, you have free and you also have bound ribosomes. All right, so they have to be exported out of the nucleus. So we have these holes there, and so they can pass through. But also it allows water to enter in, or even some large molecules can enter into, right? Ions are also going to enter in there. Okay, so it's important that we have these little passageways, all right, that will allow us for things to move in and out of the nucleus. Now, as we go into the nucleus, we have another structure, a smaller little ball, like a mini nucleus. We call that the nucleolus. Okay, it is not a membrane-bound structure. It's important that you know that. All right, but within it we have our protein and our RNA, excuse me. Okay, and this is where our ribosomes are made. All right, when I say the ribosomes are made here, the parts, we have a small unit and a large unit, and when we put those together, that will create the overall ribosome structure. And those ribosomes are then ready to make and synthesize proteins. So it's like the factory. We start to assemble the parts inside the nucleolus, okay? And then we're going to export them out and ship them out, all right? So then we can assemble them, all right, later and make our proteins. That's what we're going to see here in the nucleolus, all right? And again, we don't see this structure in all the cells, all right? But definitely in cells that are going to be making proteins. Anything that's going to be making a lot of proteins. You know the functions of proteins, okay? So any cell that is going to be making those, all right, you'll see nucleolus present, okay? Sperm cells, for example, is going to be a cell that you don't see these, the nucleolus, because it's not making any proteins. All right, quick picture here you can see, all right, here's our nucleus. All right, and this is on our electron microscope. And then inside, you can see the nucleolus. I think this is a great picture here. Kind of gives you that three-dimensional picture of the nucleus, right? And you can see there's holes all over it, right? Those are our nuclear pores, so things can move in and out, right? And then on the outside of the nucleus, you can see we have some ribosome structures. Internal on the nucleoplasm, you can see kind of looks like a grayish material here, and that's chromatin. That's going to be our genetic material. It's all kind of coiled up, right? It floats around inside this area here, and then deep is our nucleolus. All right, so when we're talking about the nucleus, we can't not talk about DNA. All right, and the DNA, and we briefly talked about DNA when we were talking about nucleic acids. All right, and our DNA is made up of repeating monomers. All right, just like glucose is a repeating monomer for glycogen, our nucleotides are going to be a repeating monomer for our DNA. Okay. And when we talk specifically, we talk about DNA being the deoxyribonucleotides when we're talking about specific types of nucleotides. All right. So to quickly just go over what a deoxyribonucleotide is made up of is we have a 5-carbon, not a 6, but a 5-carbon sugar. All right, that's going to be the deoxyribose. All right, we're going to have a phosphate molecule, and then we're going to have one of the 4 nitrogenous bases. All right, adenine, cytosine, guanine, and thymine. All right, and with all these different components here, it gives us several different variables, so you can have a wide variety and a diverse DNA or genetic makeup. Okay, and so we'll see here, all right, how these deoxyribonucleotides will be connected to one another. Okay, how we connect them to one another is through, remember we were talking about how they have a phosphate molecule. They are going to be connected to each other by a phosphodiester bond, right? I know we're getting into a little bit of chemistry. You can't help it but get into a little bit of chemistry when you talk about DNA, right? And so when we talk about DNA in regards to its structure, right? Each of those nucleotides will be linked to each other, and they'll create what we call a complementary strand. Right? Certain DNA molecule, certain nucleotides will bind or link to specific other types of nucleotides, and that configuration creates what we call the spiral ladder. Okay. And if you know anything about a ladder, you have struts and rungs. All right, rungs are the things that go horizontal. All right, and so when we pair up these nucleotides, the bases here, they form the rungs. Okay, and then the remaining part of these molecules are going to be the sugar and the phosphates, and those create what we call the struts, the side to side. So if you look here at our picture, okay, showing you here are the rungs. All right, so this is our complementary base pair, meaning guanine will only link or connect to cytosine. Okay, and adenine will only connect or link to thymine. Okay, it's different when we're dealing with RNA, but I'm not going to get into that. That's it. If you want to do genetics, then you're going to have to do that. Can you close that door for me? We're not. Okay, so we have the rungs here, and then our kind of backbone here, all right, that's going to be our sugar and phosphate group, and it creates the actual side portion. All right, so this is the DNA molecule. All right, but what happens is you have millions of genetic variables and traits and all that. So that's a lot of information, you know. And as you know, a cell is a very small structure, okay? And all right, a nucleus is even smaller than that. So we've got to get all that stuff packaged up inside, all right? And your cells have come up with a solution to that, right? Where they basically wrap up the DNA, or then they take that wrapped up DNA and then they coil it around, all right, these proteins called histones. They look like a hockey puck, essentially. And they just wrap it around tightly. And then they coil up, all right, those histones. We call those nucleosomes, and we wrap those up. So specific, I'm not too worried about the specifics, but really what I want you to appreciate is your body has figured out a way, all right, to take all this material, all this information, and tightly wrap it up, all right, and package it away to where everything about you, all right, that you can actually know from looking at yourself, and stuff that you don't even know, all right, is all wrapped up into each individual cell. It's amazing. It's amazing. So this is going to hold and house, all right, most of the genetic material. Notice how I said most, because if you recall, okay, the mitochondria carry some genetic material. Actually, 16 chromosomes are present in the mitochondria. Okay, so that's where your remaining genetic material is located. All right, so for us as humans, we have 46 double-stranded DNA molecules. And those DNA molecules are all present inside the nucleus. Okay, so what we'll do is we'll take that double helix, we'll wrap it up around our proteins there, the histones, all right? And when we've done that, now we have what's called a nucleosome, and then we wrap that up tightly into our chromatin. Okay, so like it says here, when the cell is not divided, all right, that DNA is going to be clumped into a mass called chromatin. When it does divide, all right, that tightly bound or wound DNA, all right, we call those chromosomes. And we'll talk more about that, not in this semester, but when we get into the reproduction, we'll talk about that at a later date into 11. All right, almost done here. Let's just kind of wrap things up about DNA and genes. All right, so just to review those segmental units, all right, of the nucleotides here, they are going to give us, all right, specific information to provide for the synthesis, the creation of specific proteins that, for example, could make up the color of your hair or the color of your eyes. Okay, so when we have to make these proteins, we need to know when to start the production of it and when to end it. Okay, so these DNA molecules will contain, all right, what we call a promoter region, which is the start signal. And they'll also contain, obviously, when it's time to stop, all right, making that protein, which is our stop signal. Now, again, if you were to take a genetics course, you will definitely talk more about this, okay? But we're not going to in our course. We've got other stuff we got to talk about. So I can't talk about the cells without talking about cell division. Now, cell division is going to be quite extensive. But guess what? This is the only slide. So this is all you're going to have to know for my class. Okay? So when we talk about cell division, we start off with one cell and it divides into two cells. We refer to those two new cells as daughter cells. Okay? We need cell division to occur, all right, as we grow. Okay? So when you're growing from an infant into a child into an adult, all right, we need it for tissue growth. Also for development, all right? If you damage cells, all right, we need to replace those old cells, all right, as they get too old or if they get injured. So there's two types. It's real easy. I always tell folks, start with the second one, meiosis. All right, meiosis is the division of our sex cells. Okay, so when we talk about sperm and when we talk about the oocytes or the eggs, that's meiosis. And then mitosis is all other cells, okay, which we refer to as somatic cells. Okay, so meiosis is sex cells, mitosis is all the rest. which is going to be the somatic cells. And finally, what is the function of nuclear pores? They allow the passage of molecules into and out of the nucleus. Well, folks, that wraps it up for this. I hope you enjoyed it. We will be moving on to tissues now, and pretty much going over what we talked about in lab in a little bit more detail. Like always, if you have questions, please contact me, email me, what have you. And I look forward to seeing you in class. Have a good day.